So my name is Richard Burkles, and Ian asked me if I would be the chair. Oh, sorry, I'm doing it wrong. Asked me if I would chair his inaugural lecture, and I gladly accepted. So I have to say a few words to introduce him, which I will do, and then I'll hand over to him. So as far as I know, Ian is a Brummie. Maybe even a peaky blinder. Born in Birmingham and went to school in Wolverhampton. But his father, his dad, who's here somewhere, I think, hello, worked in the armed forces. So he spent quite prolonged periods abroad as well. I think Oman, Norway, Malta, maybe other places, I don't know. But anyway, but he came back to the UK to go to university and went to the University of Kiel and studied biology and biochemistry, and graduated there not all that long ago, it feels like, to be honest, about 2001. He then landed a really cool job as a research assistant at the Sanger Centre near Cambridge, at a time when the field of genomics and bioinformatics was really exploding into life. And he was part of a team who made some really significant contributions to science. And I think this is in terms of sequencing the genomes of protozoa and bacteria of medical and veterinary importance, have been the first people in the UK and in the world to do this. And I think that I had a look earlier on, and I think there aren't many research assistants for whom 3 of their first 4 papers were in Science or Nature. So hats off to that. Ian moved to Liverpool to the Centre for Genomic Research in 2007 to start his PhD. So he did things a bit back to front. He did his PhD on bacterial symbionts of tsetse flies, and was looking at these symbionts in the context of using them to try and control the transmission of trypanosomiasis, which again was fairly cool stuff to do. And he completed his PhD 2011 and stayed on at the CGR for a few years, really waiting for the call from Salford. So he joined as a lecturer in genomics in 2015, and really since then has really kind of tirelessly worked to create and sustain and grow genomics facilities, bioinformatics capacity at the university at a time when these sciences are becoming have perforated the whole of medicine and beyond. So future proofing us, I think, in these absolutely key disciplines. For reasons unknown, for reasons unknown to me and only known to himself, in 2018 he agreed to be the Associate Dean Research for the old school of ELS and got sucked into the unification and retained the role when it tripled in size, or probably more than that, when we unified to become C. And I think that despite a changing cast list at the top of C, Ian has worked diligently and creatively since then, really to not, I'm kind of out of the limelight as well, but really been a key component of some of the major achievements of the school and the university. We can think about the climb up the ref table. We can think about the foundation of the North of England Robotics Institute. We've seen the opening of the Larkhill Brewery and the old fire station, of course, which I think is particularly close to Ian's heart. But I think also we need to recognise that Ian is a champion of young scientists here and is committed behind the scenes to facilitating the career development of the quieter voices at the university, and that shouldn't go unnoticed. Personally, I've been lucky enough to work with Ian for about the past 10 years. And we're still doing some pretty cool stuff together, but most importantly, it's been a lot of fun. I think, despite his appearances, he's quite a nice bloke. He doesn't snore as loudly as me, which works well, particularly in Uganda, although I no longer trust him with a menu in a curry house. So if ever you find yourself there, make sure you choose your own food. Anyway, I'll shut up now, and I'll present Ian to give his, and I'll invite Ian over to give his inaugural lecture.
Thanks, Richard. Ask me about cheese curry at the end. Yeah. So I've titled this From Gen X to Next Gen. So if you leave here today with anything, I hope it's an understanding of how the technology that's underpinned everything that Richard has been talking about has developed in time and in cost and in ability, and how, and I framed it around the, my career as well, and how that's sort of moved alongside it really. So after that great introduction, I hope I can live up to it. What is this topic all about then? We're talking about DNA sequencing. Well, what we're talking about is genomics. And it's distinct from genetics in so much that you're not studying individual genes one at a time, but you're studying the entire component of the genetic material within an organism all at once. So you'll start with some sort of sample. That can be blood, that can be poo, that can be whatever you like. Put it through a sequencer, which we'll come to later, generate an absolute ton of data, and then bang your head on a computer working out how it all goes back together. Again, the laughs in the audience are from people that have done it. So this really is at the interface of biology and computer science. Tell that to me in 1997 when I went to Kiel, as Richard said, and I was offered in clearing, because I didn't do so well at my A-levels, a degree in computer science and biology, which I turned down. I then proceeded to do biology and biochemistry, as Richard said, and acutely also avoided the parasitology modules. So as a molecular parasitologist, I've made some brilliant choices. Hopefully, you will remember what a DNA molecule looks like. If you don't, you'll probably have seen some sort of program on the telly that represents it as a double helix like this. If you split those two strands out, they are identical in so much that they're made up of A's, T's, G's, and C's. These are biological chemicals that the nucleotides, as we call them, and they uniquely pair up against each other. So one strand will always replicate the other one exactly the same way. So an A always partners a T, a G always partners a C. And so if you strip that strand away, you can always get back to it because it has to go back the same way. DNA sequencing has been developed utilizing this biological mechanism. When DNA replicates itself, it has to follow that process. It needs to copy itself into the next cell as that cell divides, as you develop as an organism, or as things procreate, so on and so forth. We call this sequencing by synthesis. We strip this strand away, and people cleverer than me, and indeed people that had a Nobel Prize from developing this, a guy called Fred Sanger did this back in the 70s and 80s. Here's the free nucleotides here. They've made modified ones that glow if you fire a laser at it. Brilliant idea. So you color a T green, you color an A blue, you color a C red, you color a T something else, and you put one nucleotide in at a time, and you watch it as it recapitulates. So you put one in, you read it, you put another one in, you read it, you put another one in, and you read it as it goes. So-called sequencing by synthesis, base by base by base by base by base. Hopefully that's a reasonable snapshot of doing that. That guy, Fred Sanger, had a whole institute named after him for when the general public started by it's great, isn't it, mate? Started in the US, but the US basically said, or a particular set of people in the US said, we're going to do the human genome and we're going to privatize it. The UK and Europe went, no effing way are we having any of that. We're going to set up institutes around the world to sequence this quicker than you can and put it all out into the public. So the Wellcome Trust, the primary funders that for the UK arm, set up this to sequence the human genome back in the, I think it was it must have been in the 90s by then. And I set up, this is the campus where I worked straight after my undergraduate. There's the road, there's the lake. My office, my first office was that one there. We put the sequencers there, right next to this lake, which floods regularly. Ask me that question later. So yeah, this is the genome campus. That's where we played football and volleyball. There was work done, but it was a lot of fun to be had. And as Richard said, I think that my time spent there really formulated my approach to science and academia. I think it can be fun. You don't have to take yourselves that seriously. This is what the interior of the labs looked like. Each one of these is a DNA sequencer. These things were built to be a factory because you're sequencing something the size of a human genome, which I will come to shortly. They were set up like factories. You had teams of people with discrete tasks in this pipeline to be able to turn around this mammoth task. They knew it would take tens of years and billions of dollars to do so. And so this is what this looks like. So you can see this sort of factory of these. Let me go into then the pace that was required and the technology that was being developed around DNA sequencing. So I've described the process here. As I said, you take that DNA sample. Each one of these here in a classic, what we call a 96 well plate in a lab. Okay, each one of those is one sample, one piece of DNA. The issue with sequencing, and I'll come to this again in a couple of slides, is you can't just put a genome in at the start and it just reads all the way through it. You've got to chop it up into lots of small pieces first, and reading the Gruffalo, chop it into lots of small pieces and read each of those ones individually. So you put one piece of the genome into one well, another piece of the genome into the next well, and read them all in sequence, sometimes in parallel. So the first sequences that you saw on that factory that I showed you earlier, okay, you can put one plate in here. Actually, you can put 4 plates in at once. There's a bed there. You might just be able to make out that it takes 4 plates, each with 96 samples, around about 4 hours, just a bit more than that per run. And each of those fragments that it can do, every piece of the genome it can do in one go is 700 of those nucleotides in one go. So we'll do 700, then you've got to put the next one on, and then the next one on, and then the next one on. Okay, so if you add all of that up, one of these sequences could do just under 1 million base pairs, nucleotides, pieces, individual bases, A's, T's, G's, and C's a day. So they set out, of course, to do the human genome. That's 3.2 billion of those A's, T's, G's, and C's long. Okay? So take that, do the maths on that. One instrument can do roughly, well, summarize it. If you take that one instrument and split the genome into what it can do per day, it should have taken around about 3,500 days to do, about 10 years. Now, I said that the US and the UK came together into this consortium to try and turn this thing round. And if you add all of that up, around about 200 instruments, why did it take so long then? And that's because of that bit that I said earlier, that you've got to break that genome up into lots of fragments. If I gave you a piece of string and you cut it into lots of pieces, you wouldn't be able to put that piece of string back together again because you wouldn't know how the ends go back together. Or I use a jigsaw analogy there. You need to do a genome more than once to identify how the bits overlap to slowly put it back together again. So you don't do any genome once, you do it 8 times, you do it 30 times for every genome. So that 3 billion, that 3 billion suddenly became, you know, 600 billion base pairs per human genome. So one reason there why it was starting to take so long. The other reason is genomes are absolutely daft. You can't put these things back together again quite so easily because the bits that you put in together, the A's, T's, G's, and C's, there's not a lot of data there to overlap to say, how does this go back together again? And then when it gets repetitive, if I give you an A and T and a U and A and a T and you and an A and a T, and you try and do, oh, was it me? Is it me? Is it me? That's so-called repeats. A gene in an organism has a function, right? That might be an enzyme that breaks down alcohol. Let's talk about my favorite subject, right? but you might have more than one copy of that gene, and so that will look identical. You don't know how that, you will never know how that goes back together again if you've broken it up into really, really tiny pieces. So you end up in the end with doing all the easy bits first. The computer can do that, puts it all back together again, and presents you with just the problems that are left. So actually, the human genome, the 92% of it was sequenced really, really quickly. The last 8%, not so much, because it presented us with this, not this. You can overlap all of this together, you can put it all together really nicely, but the reality of the situation was there were always bits missing. And it was that was what my first job at the Sanger was. I'll come to that in 2 slides' time. Thing is genomes are also much more daft than that, because there's only A's, T's, G's, and C's, but it's not as if those are balanced all of the time. That's only 4 types of data. But within a genome, that will vary in frequency. And so parts of the genome are really, 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 really repetitive, as in just have, sorry, press the wrong button, A's and T's versus, say, G's and C's. This is the normal balanced genome here, 25% of each of them. Malaria genome, which is my first project, thanks to a guy who will be introduced again later, Neil Hall. A's, T's are much more overrepresented in the malaria genome. Over 80% of it is just those 2 bases versus that. So of course, putting that together again, really, really tough. This is malaria again. This is what it looks like in reality. And actually, every dot on this plot is a repetitive part of that genome. Okay, so where you see, obviously there's a line along the middle, because if you put the genome against itself, right, it will always match itself. There's a solid line diagonally along the middle. That bit will always match that bit. It will always match that bit. But all of these bits also match that bit. Oh yeah, and that bit always matches that. That bit is really, really bad. Guess which bit they gave me first? Thanks very much. The thing is the reason why this is like that is actually biological. That's why we took our time to do this, because what's encoded in those regions is how malaria parasites avoid the immune system, right? Because it can change its surface coat. It can't change it too far because it stops being functional, but you change it enough so that the immune response no longer affects it, clears it, etc. So biology and function being very, very related. I talked about my first job. This is what it actually looked like. This is what I stared at for 4 years, putting together pieces of DNA on the computer there. This is the package that I used. It was called GAP4. Piecing these, the computer would do some of it. You'd be presented with a bit that wasn't quite so useful. I won't go into the details of what's here, but yeah, as I said, it was this guy who kicked me at football a lot, and gave me the malaria genome to sequence the first time. That particular bit of it, in fact, as well, and the trypanosome genome, which we'll come back to as well, had similar problems. And various bacterial genomes put together piece by piece. So you can see the A's, T's, G's, and C's being overlapped as we assemble the genome. This is what the reality of the first genome projects looked like. I did enjoy it, really, and as Richard said, I was really, really proud, because a lot of the genomes that we were putting out of medical and of scientific interest, and they were in very high-profile journals. I forget which ones on there, but you can see Plasmodium falciparum, the causative agent of malaria, the African trypanosome, which will come back again, Clostridium botulism, causative agent, for example. Lots and lots of things coming out of my lab, our lab, as it were. It wasn't me running it. I'll come to that in a second. I'm about to have one. Karen, my boss, calls me into the office one day. I've been doing some PCRs. It's a lab experiment that amplifies DNA, not really of importance. It says, "How would you like a trip to America?" Of course, my answer was yes. Didn't even think about it. Didn't know what was happening. Yeah, why on earth wouldn't I? The company set up on the East Coast that for the first time since the 1970s come up with a new type of DNA sequencing that was going to revolutionize the world. I went, sure, why not? What's the worst that could happen? And I flew out to be trained on this instrument here, the 454 GS20. This is what it looked like. It was awful. It was I'm sorry, Lasser, absolute batshit. If you've ever done, who's colour matched a paint in B&Q? Right, you know that machine that you put the paint in afterwards and it shakes it. Yeah, you put squirts a bit of black paint in that. Right, that's how this. You make the samples for this machine. You put some oil and some reagents into a tube. You put it into one of these machines. And you mix it up, and what it does is it makes a load of bubbles. Right, loads and loads of reactive vessels. It's important because you saw in that slide before that I had about 96 samples at once. Right, this thing basically said, took what was actually fiberglass, etched some holes into it, and turned a 96 well plate into a 1 million plate. Okay, so went from 100 to 1 million samples. You couldn't quite fill every hole in it, so that's why you only put 400,000 in there, and you make that by doing that, which is brilliant until you open the tube and it goes everywhere, and you've basically contaminated every experiment for forever, and yeah, and no one should ever have bought any of these, and in fact it's no longer for sale. So there we go. But the crucial thing is what we went for, and of course, was 96 samples per plate to over 400,000 per plate overnight. So you can see how that's starting to bring that 20 years down, right? From whatever it was, 900,000 base pairs per day to 40 million every 4 hours. So throughput being of the order here. First experiments we put through this new sequencing type were the same pathogens that we've been putting through, which is why I was asked to do it. So the things that we'd sequenced on a Sanger instrument, we put through on one of these just to check that this data type, this new data type, this new way of working would work the same way. So we put in a Streptococcus, and we put in chlamydia, actually, which is interesting, which is an isolate from the Netherlands. We expected to get this genome out, and we did in 16 pieces. Each one of these contigs is 16 pieces. That was the genome that we sequenced at the top. That was the one. So the reference genome, the one we did on Sanger at the top, the one that's complete and lovely. This one here is the new one. It's in a lot of pieces. It would have taken a bit of a while to put back together again, but that was one experiment in 24 hours. The first one had taken 3 years. Right. What was really interesting as well was actually we got a second genome out at the same time. We didn't know what this was, and it turns out that this is a chlamydia genome. This is chlamydia, which used to be, sorry, there's chlamydia there. I've got this the wrong way around, have I? I've got these two the wrong way around, I'm sorry. That should be chlamydia trachomatis, that should be Chlamydophila abortus. Chlamydophila abortus is isolated from the rumen of sheep and causes abortion. So I'll leave it to you to work out why a chlamydia sample also had stuff from up the rectum of a sheep in it. Someone just got it. So there's me a long time ago in a lab, in a broom cupboard, because turns out 2 years later, another kid on the block comes along, not me, but these fellas here, Solexa. They're based in Cambridge, span out of Cambridge University, and they had another new type of technology on the market. Only this time it wasn't on the market yet. They just released it. They wanted me to beta test it. The lab had left me at this point. They'd sent me out to the States to train on the 454. I brought it over, and so I was a team of 2 people bringing next generation sequencing to the UK, which is fun. So I went to the broom cupboard under the stairs to sequence with this one, and a guy who I trained up, Richard, hopefully he's online, went and ran the 454 for a bit, and so we tested these things out for the first time and against each other to see, how this is going to work and which one win and all the rest of it. So we were the first people to have both of these sequences at all, but certainly the first to have them in the UK. This one was equally nonsensical. That's where you put your plate in. I haven't got a picture of the plate on this slide. It goes in that door there. It's interlocked because you're firing lasers around. You don't want to blind yourself. But you had to put this tiny plate, certainly no bigger than this, into the machine and then load oil with a pipette all on there really slowly. Now, to get oil on top, it's broken. You get oil underneath, it's broken. You get oil anywhere else, it's broken. And you'll start again. So I really, really enjoyed my time in that lab. But actually, no, as we'll see now, we're really starting to get into the realms of very, very high throughput DNA sequencing. So remember, again, we started with that 1 million per day. We're now at 3 billion every 5 days. Similar way of sequencing. It's sort of just changes in modest changes in that, but changing the throughput. There's a glass plate, glass slide called a flow cell. I won't go into all the details of how it works, but this is it's a mock-up, if you like, or from an early flow cell there, where they've put different. Each one of these dots is a DNA molecule, an independent DNA molecule. It's what you saw before in a well on a plate. Each one of these is one piece of that genome. Scale out, of course, you get to that 100 million DNA fragments. We have traded something off in this, though. I don't know if you've been paying attention. But what we went from before was big fragments, 700 bases at one time, down to just 36 at a time. And that's a real challenge when you're starting to put things back together again, of course. You've got less data for that overlap. And so it changed the way in which we approach these experiments. We couldn't do things at the same level of accuracy, but my God, we could chuck them out. And quickly. And so really that started to change the rate at which we were sequencing these pathogens, these bacteria, and all and parasites of veterinary and medical importance. So that was all of the pathogen genomes we sequenced at the Sanger Institute in the 14 years that the pathogen sequencing unit where I worked was working. So 58 in 14 years. We got the first next generation sequences in 2004. And by sort of 2007, we'd knocked out another 23. So transformed the way in which this science was going. You can see some samples that you might recognize: the salmonellas, the Yersinia pestis, the plague, Escherichia coli, Clavibacter chlamydia that you saw earlier, so on and so forth. Things that were relatively easy, I admit. We walked before we could run, to develop the tools, of course, alongside all of this, setting the standards that the world then went on to adopt in terms of the data type that this sort of DNA sequencing, this high throughput DNA sequencing was churning out. And yeah, again, I'm very, very lucky to be on a whole number of papers at that sort of next generation now, second generation of sequencing. The first sort of comparator genomes for the human, like gorilla, yeast. I tell you, we keep coming back to beer. The first several genomes now per paper. The first time I showed this slide, it was one genome per paper. Right now we're at that was just one. Admittedly, that was one cancer genome for the first time. That was just one gorilla genome. But there, the pathogen genomes, that was over 100. That was 20 yeast genomes. So we've moved away now from one genome at a time to what we call comparative genomics, enabled by this high throughput, comparing genomes to each other to start to understand variation, what makes us individuals, what makes us different from others, say like the gorilla, so on and so forth. One day, this fella turns up from Sweden. My boss brings him in and says, "Yeah, take that and take that." I'm pointing at you now deliberately. That one's labeled M and that one's labeled N. Oh, no, fine, whatever. No gloves on or anything like that. Play with that one, don't play with that one. M was for mammoth, N was for Neanderthal. And he says, "Yeah, we just want to see whether or not this sort of technology is applicable to sequencing ancient stuff." I went, "Sure, whatever." Didn't drop it, sequenced it. It was mostly bacteria. Fine, gave them the data, walked away. Never thought anything of it. Bastard's got a Nobel Prize. Brilliant. And I was very, very privileged, of course, to have met the bloke and have done sort of worked on some of those early things. This technology is now working backwards, taking samples from ancient, museum specimens, drilling into them, taking the DNA and sequencing from there as well. Something that just wouldn't have been possible at the sort of rate of sequencing as the early stuff, because you just need so much data when it's all bacteria. When you just want that one rare piece of useful DNA in there, right? So applicable to understanding history as well. That was my time in Cambridge. This fella, as we saw earlier, who kicked me at football a lot, had just moved back from the US to the UK to set up this genome sequencing institute in Liverpool. My dad went, where? As a United fan. And but nevertheless, he said, "I hear you want to do a PhD," and I did. I mean, I was running my lab there at the Sanger now for 4 years. I didn't have a PhD, and I couldn't do it at the Sanger, or so they told me. So he said, "Well, hang on a minute. I'm coming over to see you anyway because I'm going to set this thing up, and I want to see what these sequences look like." Of course, he bought one of them, not the one that not the Illumina one that came to. Honestly, the day he came to the lab, this thing is hissing. Waste material all over the floor, and I use that word deliberately. I know I'm sweary, but the stuff was yellow, that color, right? There's just pools of it all around the place. I've got jars underneath it, things catching it's just coming out of all of the sides. I went, yeah, I'll have that one. And do you want to do a PhD? Yeah, sure. Yeah, of course I do. Thanks very much. But it's at Liverpool, fine. And so I moved there to work with Neil. We were the first people at the CGR, but it was to work on something that I'd worked on briefly at the Sanger called the trypanosomes, African trypanosomiasis. I can't believe I've only got 10 minutes left. This is going to be an interesting second half. So my samples from Uganda, just north of Kampala. Kampala's down there. Sequencing samples held at the Liverpool School of Tropical Medicine, as it happens, to understand the variety now within and between genomes that cause African sleeping sickness. It's a parasite spread by tsetse flies. Each one of these is an individual that had sleeping sickness from an outbreak in the 1980s, and we described for the first time genomes of Trypanosoma brucei rhodesiense. It's a particularly acute form of the disease, and we had identified that one particular strain, these match the map over there, the ones in red, were more closely related to an animal infected strain. This one in blue here, which was actually more acute in humans, had picked up some genetic material from a similar parasite called Trypanosoma brucei gambiense. And so this is a phylogenetic tree, and so the distance, the closer things are to one side than the other, shows how related they are, basically. So this is more closely related to these ones, and these ones are more closely to these ones. And this is by doing whole genome sequencing, admittedly on a slightly different sequencer again. A third one's on the market now called Solid. I am not going to go into it. Why Neil bought this one? It took all the worst bits of 454, the shaky bits, and added more craziness to it, and he gave me that to do the PhD on, but it was incredibly high throughput, which is useful for doing genomes quickly when you had something. I'd already sequenced it on a Sanger ages ago, so we had something to compare to, which meant that we could do this type of thing, but chuck out 1 billion reads in one experiment, so we could quickly get the data. It was nonsense, but some of it was useful. Catherine, my flatmate, and Jack was with me at the time as well for my PhD, sat over there, will remember me scratching my head, working, late at night, 10 p.m. on my computer, hair drifting across the. Anyway, talk me afterwards. Trying to put this data back together again. It's fun. Oops. So this is the Centre for Genomic Research. So completed my PhD there and decided to postdoc. This equipment is actually reasonably expensive, right? The Solid was about £500,000. The modern sequencers now, these are it's not quite the current generation, admittedly, but the Illumina HiSeq, which is the evolution of the one in blue that I saw you saw earlier, which is now the market leader, sold comfortably worldwide, easiest to use. We should have bought those in the first place. This is it here. You can see, you can work out for yourself to the throughput that these things have. Again, these are about 300, 400,000 pounds a go. Not approachable by every lab everywhere, right? You need centres to have these, invest in the infrastructure, and then we offered it as a service science, pretty much at cost recovery. We'll do your experiment for you, give you your data back. So I did that as a postdoc for 3 years, either doing the sequencing in the lab or indeed the bioinformatics. It was also where I met my wife, future wife at the time, because I was given her project to do the bioinformatics on. It wasn't actually this experiment. We never published that. Anyway, fine. Not bitter. But this is a transcriptome experiment of Campylobacter jejuni. You may have heard of that as food poisoning. So this is comparing gene expression. Now, how much a gene is used, more, it's red. If a gene is used less, it's in blue, and comparing that over time in environmental samples to understand how this bacteria survives in the environment. I was given that project to work on with her, and well, the rest is history. And well, you've all heard and met Lassa as well. I'm very proud of my work at the CGR, actually, and I was fortunate enough to hang around long enough that they offered me a postdoc, which really exposed me to a whole new area of science I wasn't expecting, and that's symbiosis. This is bacteria within other organisms, okay, which becomes really important. I asked some questions earlier about how these evolved. Again, if you sequence something that's inside something else, that's really, really hard, because you don't just sequence, you can't just separate it out. Well, you can, I'll talk to you about that in a bit. You take one thing out, you sequence one thing, but actually you just sequence all of it. Then you've got to put it all back. Wait, did you belong to that one or that one? Right, so putting it all back together again can be problematic. We were interested in one particular bacteria that I won't have time to go through, but it's really interesting. Again, in that trypanosomiasis system, because I was interested in. Sorry, I've just pulled that mic. Hopefully that's still on. In this idea of not just one genome at a time, but lots of genomes at a time. These bar charts here are representative of a different species in one sample. Basically speaking, if you see a different color, it's a different species in that one sample, and you can start to see that these things are incredibly complex. You've heard, I hope by now, of microbiomes as a concept, and probiotics and stuff like that, your microbiome being really important for your health. It's this technology that tells us about the species that benefit you, or when that gets knocked out of kilter, are more problematic, right? That's all this problem. And it's this technology, sequencing the genomes within that tells us exactly what that profile is. Only I'm not interested in humans anymore. I'm interested in parasites and still sleeping sickness. So one particular subject I was pleased to have got some funding for when I got here, and my first PhD student, Poppy, who's sat in the corner, you'll see her face on the poster up there, is this idea of sequencing inception and understanding how these organisms play with each other, well or without, and using that sequencing technology to do that. Sequence everything, put it all back together again, and then work out, well, okay, how is that contributing to this problem? For example, tsetse flies, the vector, the fly that causes sleeping sickness, has an organ in it that contains a particular bacterium that without it wouldn't survive, because it provides it with nutrients that it wouldn't get from its normal meal of blood. And so it's understanding how these things work together and using DNA sequencing to tell us more about the makeup of these ecosystems, really. I'm particularly interested as well now in ticks, since I've come here and worked with Richard. He's a brilliant tick microbiologist, or pretend microbiologist, and so we've been doing a lot of really fun work of understanding this in ticks as well. Poppy's work went on to do some work on the genes in that as well. But so take a fly, mash it up, and sequence every single gene in there several times, and that's from every organism in there. If I just skip back a slide, there's the tsetse fly. There's all the genome. There's all the organisms that are within there. Actually, it's not quite all of them, but a good number of them. So imagine now taking every gene within that. Remember, the size of the human genome is 3 billion. That's 25,000 genes alone. Then so the fly has genes, the bacteria have genes, the parasites have genes. Sequencing each of those, counting how many times those genes are used at a particular point. This was an experiment to understand why these flies actually are pretty normally anyway resistant to spreading trypanosomiasis. I won't give you the details of what's happened, but and we haven't published it yet. One day we will, won't we? Thank you. We also did some work on sandflies. I'm running out of time, so I'm not going to go into this. But this was sequencing individual sandflies, which are really, really tiny. Really tiny. It was a real pain in the backside. But that was, again, those are those bar charts that you saw earlier. Understanding, using that same sequencing technology to understand the makeup and the relative makeup of the bacteria within that to try and intervene in diseases like trypanosomiasis, or indeed, in this case, leishmaniasis, which is a similar disease. Hi, Judy, you're up on the screen as well. This is how we're using it right now. The reason why you understand what the COVID variants are is the same technology. Right? We've sequenced them. So an Omicron has a particular sequence, an Alpha has a particular sequence. Only we had a project to try and set this technology up. I told you about the challenge of sequencing is very, very costly. So we tried to do this in Uganda. We had global charitable money to set up a lab to study this in northern Uganda, which is starting to be applied to local problems as well. Very, very out of time. I'll go on a little bit late. It's all right. Sean is here right now working on this to pick apart this problem even further. We're interested in bacteria of veterinary importance and zoonotic importance. Zoonotic is where an organism can spread from animals to humans. But Anaplasma is a particular bacterium in various veterinary animals. The problem is it's intracellular. It lives inside a white blood cell. So you take a white blood cell, you sequence a human. An Anaplasma has a really, really tiny genome in comparison to a human. Only 1 million base pairs versus the 3 billion that you saw earlier. So we're working on ways in which we can enhance that. He's done some great work sequencing for the first time UK isolates. Our isolates here in light blue. These have been done for the first time. You can see around the world that others have been done, but no one's been looking at this in the UK so far. So that's work that he's writing up for his thesis now. Incredibly impressive. And what he's been doing is using even the next generation of sequencing to help with this. I'll come to that in my next slide. He's designing mechanisms in the lab that can basically take that sample and take out in the lab the bits that you don't want before you sequence the bits that you do. Right? And it's really, really important if we want to get at these tiny, tiny bacteria in amongst this nonsense that is a human that we don't, or a sheep or a deer or whatever that we don't really care about. So he's putting together these experiments to enrich for microbes, and this really, really science fiction thing here called adaptive sampling here. This is the 3rd generation sequencing, which is a nanopore. Okay, here's the DNA molecule going through the nanopore. Here you can see a T, an A, a G, whichever, a C there. Ignore the health and safety boss, sorry. This is demonstrating a nanopore sequencer. This thing is portable. You can plug it into a laptop, take it wherever you go. Joe sat over there and I have done a brilliant work sequencing a music festival and all the bacteria that they had. Yes, it's as bad as you think it is. Crucial part there is look how much it is. This is transformative now, 1,500 quid per device, but it also does this really cool thing that it's actually reading the DNA as it's passing through this hole. This is a microscopic hole in a membrane, okay? Imagine a cell, a really tiny cell. This is a cell wall here. This is a pore, a hole within that thing. And so there's a couple of thousand of these pores in an array that you're putting DNA in through at any one time. I'm being told I've got time up, sorry. And so it's reading it, and as it goes, it can basically say, I don't want you, I don't want you, I don't want you. So everything I said about that enrichment, the computer is doing as it's sequencing at the cost of 1,500 quid. And this thing will actually churn out enough to do very, very big genomes. This is transforming the way in which we are doing sequencing. There's that Anaplasma. We've gone from 0.0008% to 66% in some samples using combinations of these techniques. We're also working on ticks still. We've got active work right now to establish the same technology now in Mwea in Queen Elizabeth National Park. Richard and I have funding from the US to establish this sort of sequencing to work on tick-borne and emerging pathogens. You may have heard of Crimea Congo hemorrhagic fever. It's a bit like Ebola. You don't want it. But using sequencing as a sentinel program to identify when these things are emerging, so you can put programs in place to intervene early. In the same way that this is used for COVID and whatever else, and identifying at particular what was it, variants early in wastewater, for example, you may have seen in the news. So we're identifying ticks there for emerging things. Really proud of this work. Jean and Robin have done incredible work setting us up in terms of sequencing primates for the first time, and in so doing, we've had a really cool paper recently on this. Incredible scientists, and I feel very privileged to work with them and their team. We're evolving that now to use this same technology, nanopore technology, to sequence endangered species and inform conservation efforts for some of the most endangered species on the planet, partnering with the company that makes this, and of course my favorite subject, beer. The first time ever, me and Ali, who sat there in that corner, are starting to think about how can we bring science to the brewery, because I like drinking and I like brewers, and they don't do science very often. Sorry if there are any brewers online, but you don't really. You make a beer and it tastes nice, and I'm starting to think about how we can do some of this. For the sake of it, we've set up a little bit of a test lab. This is at Marble Brewery here, and we brewed a couple of beers, and we've been playing with them to how we can help them understand their beer and test. Well, how does sequencing come into this? Well, we're starting to sequence their yeasts to understand what genes bring what flavors to certain beers and things like that. And you may have heard of things like genetic modification. CRISPR is in the news. It's been a couple of films now, very famously, how you can modify genomes to do what you want to do. So you can put flavors into genes into yeast that produce particular flavors that people like, the bananas, the passion fruits, so on and so forth, then we can start to do that. So we're setting up a lab this year, hopefully to start testing around this, and our first project will be sequencing the Manchester yeast biobank, which we're going to set up here, which will be fun if we get money. Penny, hi, thanks. That's me, everyone. I'm just going to wrap up by bringing this back to this idea of what that throughput means to us. No genomics talk would be complete without presenting this slide, which is the cost per human genome and how it's evolved over time. It was originally compared to Moore's law, which is the idea that computers would double in speed and half in cost every year. So that's that straight line down there, and that was the cost back at the start of the human genome back there, around about $100 million. So 2001, when it was published, you can see where second generation sequencing came in, and the cost per genome absolutely fell off a cliff. I had it in 2004, just saying. And then proceeded to come down. So we're now at this inflection point here, around about $1,000 per human genome, which is pretty impressive and is really close to frontline care. And it's actually the partnership of these technologies and, coming to me, and NHS that allows for us to be able to do this sequencing close to the clinic. Right, these are starting to be set up. NHS Genomes England is starting to bring this really close, because it's at that $1,000 per genome. It's actually getting towards the point where rare diseases can be diagnosed using this technology. Cancers can be identified and sequenced, and tailoring treatment to a person's individual cancer, personalized medicine, as a consequence of the improvement in sequencing and the reduction in cost therein. So really the question we have to ask now is, this is going to come within our lifetime because of this development of this technology that you've seen, and we have to ask those questions of what will we do now that this is accessible. It's almost portable as well. You've seen they can take this around the world, so it's really changing the landscape of biology and medicine. I hope that's been of interest. Thank you very much.
Right, well, thank you, Ian. Very interesting, big numbers, very scary. I've got some questions from the online audience, but equally, if somebody in the room would like to ask Ian a question, now is your opportunity.
Stunned into silence. It's just like my lectures.
Robin, you're first.
An early question.
Yeah.
So when you sequence plasmodium,
Yeah.
why couldn't you sequence chromosomes?
Why couldn't we? Oh,
Yeah.
we could. Yeah.
Yeah.
Yeah. Great question. So, Robin, so those of you that are at home who might be able to hear that question, he's pointing to this point here, which is when we sequenced the malaria parasite, why did we only do 1, 3, 9, 3, 2, 9, and 13 in that experiment? They were all done separately. The way in which this was sequenced is it was separated chromosome by chromosome. So there are mechanisms in the lab where you can put DNA into what we call an agarose gel. Okay, so it's a matrix that separates out DNA according to its size. Okay, so chromosomes, which I hope you understand, separate out at different paces. You can cut them out at that point and just sequence that bit. There's a way of simplifying the problem of rather than doing it all at once. So the project was separated out into those chromosomes and done chromosome by chromosome. Some were easier to do than others and were therefore published at different times. And I was given the blob, which was a combination of chromosomes 6, 7, and 8 that when you separated it all out, they were so close in size that you couldn't distinguish them. So you just had to do them all at once. And so it's hidden in amongst all of this.
Okay, one from online. I think I know the answer to this one. Given how much it costs to sequence, have you ever messed up?
What have I messed up? Oh, I mean, first time we got this, 2004, brought it over, put the first sequencing on, orange screen of death. The very, very first experiment we ever did on a next generation sequencer in the UK failed. Yes. I want to skip to, I mean, I don't even talk about the oil. My God, I want to skip to this slide, this one. I mean, what might not come across in all of this, which is my fault really, is these are all well and good, right? 500,000 per instrument and $1,000 per genome. But to achieve that level of throughput, you've got to put on a lot of genomes at once. Okay, you don't just put on a genome for $1,000. You put on 24 for 24,000 or 48 for $48,000. The throughput, the paradigm has changed, right? Away from just being able to do one genome in 20 years to doing, yes, fine, I can do 20 genomes for you in 3-4 days now, but you've got to give it 20 genomes at a time. So you've got a lot in the lab, you've got a lot at the analysis. And sequencing is pretty easy, but the sequencing still costs £25,000 per run. And the plates are made of glass. Yeah, I have £25,000 worth. Dropped at least one. I also, at one point, got the very last point of it. You've got 2 tubes to mix together in different amounts. One, you remember that bit I said you've got to take those DNA strands apart with my very first slide. One of those does that. The problem is if you put too much of it, they'll never go back together again. And I got the 2 tubes mixed up, so that didn't work. £25,000. I once did the maths actually when I started when I started here at Salford. I gave a talk obviously on this subject, and I'd actually added up how much money I'd wasted, and it was over £500,000. So yes, yeah, I have.
Greg, did you have a question?
What do you think is coming next? I kind of remember from about 10 years ago, there was a guy called Braxton who was talking about a nanopore thing, but without the translocator, just being able to sequence. And obviously it hasn't worked for him because he reckoned that this would blow all this away.
Yeah.
And he perhaps snowballed his career.
Right now, we're in a phase of understanding. Right, we can chuck these genomes out. We still don't understand what they say. AT GNC is all well and good, But what does that mean? Actually isn't at the DNA level, it's at the protein level, it's at the enzyme level. So we're in a phase now of analysis and understanding. AI will come into that, machine learning will come into that, understanding the impact that this has on human health. You know, if I can sequence you and I sequence you and I sequence you, and we say, well, okay, what does that actually mean for me and my health? Right? We're not necessarily there yet. There's something being developed called what we call polygenic risk scores, right? The idea that anything that you have isn't just one gene, it's many genes, polygenic. And so you have a risk associated with some sort of thing that you might have, which has to be the quantitative balance of that genome, right? So you need to develop tools to do that, and AI and machine learning is developing that. But your question, I think, was more aimed at the technology as well. This is biological right now, which means it burns out after 24, 48 hours. It's derived from a bacterium. It's a natural pore by which bacteria take up nucleotides so they can reuse them, right? It's cool. So it's been co-opted for that. Synthetic versions of that are coming. The next technologies will be so-called solid state nanopores that won't burn out, and thus, in theory, could just be reused and reused and reused and reused, and thus would drive that price down even further.
Okay, I've got a question from someone online who says, which of the technologies have you spoken about do we have at Salford University? And with the PVC research listening, if you were offered the opportunity to buy some new technology, which would you choose?
Okay. Actually, thank you to the university for providing us with quite a lot of equipment. We've got one of these, it's an aluminum MySeq, which is kind of a benchtop version of a sequencer. It's enough to do a number of bacteria all at once, but can't quite do a human sort of. By then. And we've got this. That's not my hand, honest boss. But we've got two of those, one of which we use for research and one of which we use for teaching. Hi. We're very proud, of course, to be building this into our teaching programs, right? We want to be teaching our students this, so we've got one specifically for training our students on. We've also got one of these. This is a P2 Solo, which is for human. We can do humans, we can do big genomes, we can do the endangered species that I talked about earlier. That's come out of recent funding out of the research knowledge exchange here, thanks to Janet Marana for that. Yeah, that's it. If I could have anything, actually I'd stick with that, because I don't want to deal with the data, the amount of data that comes out of. Hi. Oh yeah. I don't want to deal with the level of data that comes out of one of those machines, right? That's the problem. It's all good generating the data, but analyzing it is really hard. So I'd rather give that to someone else.
Okay, I think Lasser is saying he's had enough.
There was a.
So can I ask one last question?
Go on then.
So I love prosimians, but from a genomics perspective, what's your favourite monkey?
Wow, that's my favourite monkey. Hi. Is it this one?
Okay, well, thank you very much for coming. Thank you for all of you who've joined us online. We're going to make a small presentation to Ian now, but I think the people online are leaving us now, so thanks for joining us.